Imagine trying to reconstruct a crime scene after a bulldozer just drove straight through it.
Right. Impossible.
Yeah, but if you're looking at early-stage bladder cancer, I mean, for decades, that is essentially what the standard surgical approach has been doing to the tissue.
It really is.
Welcome to the deep dive, by the way. Today's mission is to uncover the exact mechanisms of how early-stage bladder cancer learns to invade deeper into the body. And you probably know the basic statistics already. Bladder cancer is the 9th most common globally, heavily driven by urothelial carcinoma.
Yeah, and the real clinical bottleneck is that while, 75% of cases present as non-muscle-invasive bladder cancer, or NMIBC, up to half of those will stubbornly progress into muscle-invasive disease.
Exactly. And we need to know why and how that happens. So today, we are skipping the basic definitions and digging into a groundbreaking 2026 study from Nature NPJ Precision Oncology. They basically map out this progression line by line using spatial transcriptomics to uncover the literal machinery of tumor invasion.
And what makes this specific study incredibly rare is the tissue itself. They used strictly treatment-naive tissue.
Oh, meaning no chemo beforehand.
Exactly. We are seeing the cancer in its natural state, undisturbed. It's before any chemotherapy or targeted radiation has altered the environment or triggered massive cellular die-offs.
Okay, let's unpack this. To get that uncompromised look, the researchers had to ditch the bulldozer we talked about. I mean, you're familiar with the standard TURBT procedures, right? The transurethral resection.
Blender approach.
Yeah. It's like taking a beautifully layered cake and throwing it into a blender. You get the tumor out, sure, but the electrical loops fragment the tissue. They cause severe thermal burns.
The whole 3D architecture is just ruined.
Totally destroyed.
Yeah.
Pathologists are left guessing about the depth of invasion based on this pulverized sample.
Right, but the clinical shift that makes this new spatial analysis possible is ERBT. That's en-bloc transurethral resection.
So no more blender.
Exactly. Instead of scraping it out layer by layer, the surgeon meticulously traces a 5-millimeter margin around the tumor, dissects down into the superficial detrusor muscle, and removes the entire malignant mass in one single intact piece.
So keeping with the cake analogy, ERBT is like using a sharp knife to lift out a pristine slice.
A perfect slice.
You see the frosting, the filling, the sponge, all locked in their original spatial relationship. And because they preserved that map, they could run 10x Visium spatial transcriptomics on it.
Which is huge, because traditional bulk RNA sequencing, it gives you a list of active genes, but it strips away all the context of where those genes were active.
Right.
The 10x Visium platform solves this. They lay that intact ERBT tissue slice over a highly specialized microscopic grid.
And this grid has thousands of tiny spots, right?
Yeah, and each spot is exactly 55 micrometers across. Every spot is loaded with probes that capture and barcode the RNA from the tissue directly above it.
Wait, I have to push back on this a little. If a single spot on that grid is 55 micrometers wide, that holds roughly 10 individual cells. So if you pull RNA from 10 completely different cells simultaneously, how is that not just a molecular smoothie? I mean, how do you know which cell is doing what?
That's the brilliance of the deconvolution tools they used. They didn't rely on the hardware alone. They applied algorithms, specifically SpaceEta, STD-Involve, and RCTD.
Okay, and what do those do?
They mathematically unmix the smoothie. They use massive single-cell RNA sequencing reference databases. And by mapping the blended RNA signature of that one spot against known profiles, they calculate the exact cellular proportions.
Oh, wow. So the algorithm can look at a spot and say, okay, this is exactly 60% malignant cell, 30% CD4 T cell, and 10% stromal cell.
Down to the decimal. Yeah. And that deconvolution reveals the tumor microenvironment, the TME. You map the precise location of the immune cells, the CAFs, the endothelial cells, all while preserving the 3D map.
Okay, so let's do a quick recap here. Bullet 1, bladder cancer often starts superficial but frequently turns invasive.
Right.
Bullet 2, ERBT surgery gives us an intact 3D tissue slice, unlike the old fragmented method.
Yeah.
And bullet 3, spatial transcriptomics lets us map exactly where genes are active on that 3D tissue.
Perfectly summarized.
So think about it. If you are a cancer cell trying to invade, where would you set up your operations? Let's look at the map.
Well, the study analyzed 7 high-grade PT1 patient samples. And when they mapped the tissue, they found the cancer divides itself into 2 distinct zones, the tumor upper region and the tumor-boundary region.
The boundary being the invasive-front, right? The trenches.
Exactly. And the uppy region, the part facing the inside of the bladder, functions essentially as a biological factory.
Churning out cells.
Relentlessly. The spatial map showed massive upregulation in MYC targets, oxidative phosphorylation, and E2F targets.
So MYC and E2F force the cells to continuously divide, and the oxidative phosphorylation generates all the ATP needed to fuel that factory.
You got it. It's just nonstop proliferation.
Here's where it gets really interesting, though, because the boundary region is entirely different. It's not focused on dividing. It's focused on moving.
Yeah, the genetic signature completely flips. At the boundary, you see a high enrichment for EMT epithelial-mesenchymal transition.
The cells are changing shape to become mobile.
Exactly. Plus massive spikes in focal adhesion and PI3K ACT signaling. The cancer cell has to bring its anchors, shift its shape, and crawl. And the PI3K ACT pathway provides the survival signals so the cell doesn't just die when it detaches.
But the boundary isn't just cancer cells, right? I mean, the deconvolution showed it's teeming with immune cells, B cells, CD4 T cells.
Which you would think is a good thing.
Right, you'd assume the body's fighting back. But there's this deceptive communication loop called light signaling.
This was fascinating. The immune cells are actively secreting a signaling protein called TNFSF14, which is known as light signaling.
Okay.
But the receptors for that signal, TNFRSF14 and LTBR, are profoundly enriched on the surface of the tumor cells right at the boundary.
Wait, really? So the tumor cells are deliberately catching the signals from the immune system?
Yes. And when that LTBR receptor gets activated on the tumor cell, it triggers the non-canonical NF-kappa B pathway.
Oh, so instead of being destroyed, the tumor cell uses that immune signal to upregulate its own survival genes.
Exactly. It's weaponizing the inflammation to adapt and survive.
Which perfectly lines up with the clinical cases in the study. Because out of the 7 cases, 3 of them recurred patients, S2, S3, and S4.
Right.
And when they looked at the boundary regions for those specific recurrent cases, they found 107 upregulated genes linked to chronic inflammation, cellular dedifferentiation, and therapy resistance.
The tumor is literally feeding off the immune response.
It's wild. But to really understand how dangerous this boundary is, we have to talk about the physical barrier, the muscularis mucosae.
The ultimate line in the sand?
Yeah. If the tumor is superficial to that thin layer of smooth muscle, it's substaged as PT1A. There were 2 of those in the study.
But if it invades beyond it, it becomes PT1B, which is much worse. 5 cases had breached that line.
And PT1B has a significantly degraded progression-free survival rate. So what was happening at that specific PT1B boundary?
The boundary in those PT1B tumors was buzzing with cancer-associated fibroblasts, or CHAFs. And the spatial data showed the tumor cells secreting high levels of midkine, or MK, and pleiotrophins, PTN.
Which act on the CHAFs.
Right, exactly. The CHAFs respond to those signals by going into overdrive and totally remodeling the surrounding matrix.
Okay, so the researchers wanted to find the exact genetic blueprint for this. And they extracted 15 specific genes that are upregulated in PT1B tumors, A and D, highly enriched at the boundary.
The 15-gene signature of invasion.
Yeah. And to remember how these 15 genes work together, I like to split them into 2 groups. Group 1 is the paving crew.
I love that analogy, the ECM remodelers.
Right, so the paving crew is genes like TIMI2, POSTN, CCOMP, FBLN5, LEOM, and CDH11.
So what they do is they dump excessive extracellular matrix, the ECM, and they heavily cross-link it.
Basically turning soft tissue into a rigid highway.
Exactly. They make the ground stiff.
But a stiff highway isn't enough, the cell has to move down it. Which brings us to group 2, the engine.
Genes like DES, ACTG2, and MYH11.
These promote myogenesis and contractility, right? Actin filament polymerization.
Exactly. The engine gives the cell the mechanical tension, the actin-myosin contractility, to grab onto that stiff highway and physically pull itself forward.
It's synergy. The paving crew makes the road stiff, and the engine cranks the cell forward to invade.
It's brilliant mechanics.
Okay, time for another quick recap. Bullet 1. The tumor divides its labor into a factory upper half and an invading boundary half.
Yeah.
Bullet 2. Tumors that cross the muscularis mucosae into PT1B are far more dangerous.
Definitely.
And bullet 3. A 15-gene signature at the boundary drives this by stiffening the tissue and pulling the cancer cells forward.
Spot on.
So if we know exactly which 15 genes drive this invasion, how can we use this to save lives? Let's look at the bigger picture.
Well, if we connect this to the bigger picture, we have to remember the Visium spatial tech only covers a tiny 6.5 millimeters square.
It's a very small snapshot.
So to prove these 15 genes matter globally, they tested them against massive public bulk RNA-seq databases, the Romal cohort and TCGA-BLCA.
And what did they find?
Unbelievable results. High expression of that 15 gene signature means significantly worse overall survival and worse progression-free survival.
Wow.
And it scales up with the clinical severity. The score increases from T1 to T4 stages, and it's significantly higher in patients with lymph node metastasis.
Okay, but let me push back a bit here. If we know the proteins these 15 genes make, why don't we just use standard hospital staining, like normal IHC, to look for these proteins under a microscope?
It's a great question, and the researchers actually tried this. They stained for 5 of the key genes. The proteins were there, but the visual intensity didn't look different between PT1A and PT1B to the naked eye.
Wait, really? So IHC isn't sensitive enough?
No, the danger is really only visible at the RNA transcriptome level.
But we can't run a wildly expensive spatial transcriptomics test on every single patient. That's just not feasible.
And we won't need to, because the ERBT surgery gets that boundary completely intact, right?
Oh, right. The pristine cake slice.
Exactly. So hospitals could eventually just run a simple, cheap qTCR panel strictly on that boundary tissue.
Just checking for the RNA of those 15 genes.
Yep. To catch that signature and predict if a patient's cancer is gearing up to aggressively progress.
So what does this all mean? We preserve the geography of the tumor with ERBT. We use spatial transcriptomics to map it out, and we reveal the literal machinery of cancer invasion at the border, the paving crew and the engine.
Which raises an important question for the future of oncology.
What's that?
If cancer invasion relies so heavily on building this stiff highway of extracellular matrix, what if the future of therapy isn't just about attacking the cancer cell, the seed?
Oh, I see where you're going.
What if we develop drugs that soften the tissue around it, the soil?
Soften the soil so the seed can't spread. If the paving crew can't lay down the concrete, the engine has nothing to pull against.
Exactly. The cancer cell just becomes physically trapped.
That is a fascinating paradigm shift to think about. Thank you so much for joining us as we unpack this incredible study today. For you listening, keep that concept of softening the soil in mind, keep questioning the world around you, and we'll catch you on the next Deep Dive.